for coming out. This is our fourth Innovation 101 Capitol Hill briefing, and we started these with the intent of helping staffers, administration officials get a first-hand view of what innovation really is like, what it means to have a startup company grow, what it means to have research, development, and commercialization. We think that the best way for you all to learn about that is to hear it firsthand from the people that are doing it. We have a great lineup today with Sanford Burnham and then a startup company that is actually trying to commercialize research. And so in addition to all the challenges that a startup company has just making it into the marketplace, there's all sorts of other policy, regulatory, and financing hurdles that they go through. And so our intent with these briefings is to give people a real-life view of what is it like to try to take research, commercialize it, and what's it like as a startup company to actually break in and make some progress in the market. San Diego is home to a thriving innovation economy, a thriving innovation hub, and we start with the core, which are our research institutes. As you can see, we have Sanford Burnham up there. University of California, San Diego is a leading research institute. The Von Liebig Center is an incubator that helps these companies grow. And then SPAWAR is in the defense sector, the leading research lab on the West Coast for the Navy. So it's over 83 research, up there it says over 70 major research institutions. The latest count I've seen is that there's 83 research institutes. By far and away, research institute employment outpaces the rest of California. It's by 10 to 15,000 more employees in San Diego research institutions than the rest, any other area in California. And so we start with our research institutes, but we are very diverse in the technology. This is our fourth Innovation 101. This one we're focusing on life sciences. Before we focused on proof of concept centers and incubators. The time before we focused on algae and biofuels. And before that we started with Bill Walton, famous NBA basketball player who works for Connect as our head of sports innovation. So in San Diego, you're seeing all sorts of surfers, bike riders, athletes realizing, hey, I can make a better surfboard, I can make a better helmet. So there's just a thriving innovation community, and everyone in San Diego seems to have this entrepreneurial spirit that thinks, hey, I can do that better, I can do this newer. So we start with defense and security. There you have SPAWAR, the unmanned drones that you hear so much about. Most of that technology and that development occurs in San Diego. As I mentioned, IT, wireless, and software. Qualcomm, one of the leading chip makers, is based in San Diego. And there's our strong life sciences community. Our startup is Sendar, and they're up there on our map. But the biotech industry, some have said, was birthed in San Diego. So back in the 70s with Ivor Royston and Howard Bromdorf. And then energy and environment, because of our great climate, we've got a lot of solar production, but we're getting into everything, like we said, biofuels, other forms of alternative energy. And then there's our sports innovators. You can see all the dots represent companies. Connect was formed 25 years ago out of the University of California, San Diego, with the sole purpose of helping commercialize research and innovation and help startups grow. 25 years ago, the founders of the city leaders in San Diego said, we've got a lot of great research going on, there's a lot of great papers here, but we're not getting companies, we're not getting jobs. And so Connect was created to help those people take those steps necessary to start their companies. Last year we won the Economic Development Administration's Innovation Award for Regional Economic Clusters. And this is one of the key data points that I wanted to have you all take away. As Congress focuses on what should we be doing to create jobs, we argue that Congress should be making its attention on tech sector jobs. And here's why. In San Diego County, tech sector jobs are 6% of the employers in the tech sector, only 11% of all jobs. They make up 25% of all the wages, but those wages are 114% higher than the average wage. So and it's a little hard to see, but you have the average wage in San Diego, non-tech sector, is $45,000. The average tech sector wage is $97,000. So not only are these highly intelligent, highly skilled individuals, they're getting paid more. There are jobs that go with those high-skilled jobs, and that has a huge economic impact. And you can see throughout, regardless of the technology, those jobs are all outpacing. So as Congress thinks about their jobs agenda, what might be able to get done in this difficult election year, we really think the focus should be on tech sector jobs. And what we see today is Sanford Burnham, a leading research institute, especially as it relates to NIH funding. They're one of the top 3 research institutes with NIH funding, is helping to develop research and then keep it moving and create those jobs. So without further ado, I'd like to introduce Dr. John Reed, CEO of Sanford Burnham. They are a bi-coastal organization. They have facilities in California and in Florida. They pick really difficult places to have employees, but and so without any further introduction, please welcome Dr. Reed.
Great. Well, good morning, and thank you for taking time to attend this session and to learn a little bit more about life science technology innovation. I'm going to focus most of my remarks on the role that NIH plays in terms of funding the fundamental research that creates a whole ecosystem of economic development opportunities that drive the biotechnology and biopharmaceutical industry. And I think the context in which to look at this is recognizing that it's been touted that biotechnology will be to the 21st century what information technology was the last century. So when we think about creating jobs in this country and we think about global competitiveness, this is the area we really need to pay attention to. And I'll tell you a little bit about what an important role that the nation's investment through NIH funding of medical research is making. If you don't get anything else from my talk, this is the take-home point, which is that the investment we're making as a nation in medical research not only is playing an important role in improving healthcare outcomes, but it also is creating the foundation for economic development. So it's both health and wealth. I'm going to try to touch on 3 different points. There are 4 listed here. I won't have time to go into all these, but I want to talk a little bit about the sort of ecosystem that exists in terms of how basic research creates the discoveries that generate intellectual property, that becomes the substrate for creating new companies that attract capital and produces jobs and economic growth. That's the first topic I'll talk about, and talk about the role that NIH funding plays in that. I'll also talk about at a global competitiveness level, about how the patents that we generate from the discoveries that are made in this area play such an important role in positioning the country for economic success. I won't have time to talk about the important role that NIH funding plays in creating workforce development, and that is in the training of the scientists who lead this life sciences enterprise. This is a very important aspect of what NIH also does, and it's something that we should be really paying attention to. It's been, with the current situation where NIH budgets have been effectively flat or in decline when adjusted for inflation since 2002, and where other nations are really ramping up their commitments to and investments in medical research. What we're seeing now is what's been referred to as a broken pipeline in terms of encouraging our best and brightest to pursue careers in this area, as well as a brain drain in the country where some of our best and brightest are now leaving the United States to go work in other places. So it's something we should really be paying attention to, but I won't say any more about it today than that. And then finally, I want to talk about healthcare costs and the role that medical innovations play in driving down healthcare costs and trying to make the practice of healthcare more efficient. That's something, of course, we all care about as we look at the rising and mounting costs of healthcare. So just to set the stage here in terms of looking at this ecosystem whereby research leads to new innovative medical products, I just need to educate you a little bit about the pipeline concept of how basic discoveries that reveal causes of disease mechanisms, help us to identify the targets that we want to pursue with new medicines, and then lead to the prototype, what we call lead compounds or first attempts at trying to address that, new medicines. And then they move down a process, a complicated process of phase 1, 2, 3 clinical trials. Hopefully the FDA eventually approves them, which is a whole other conversation. And hopefully then become commercialized products that create revenues for the companies that have invested in these, which contributes to our tax base, of course, but also products that help to improve quality of life and healthcare outcomes. Now, the NIH, which funds about 80% of all the basic discovery research in the country, plays an important role at these early stages. These are places where companies really don't invest. And so that discovery research creates a foundation that then is important for, and indeed essential for, the rest of the downstream pipeline. Altogether, we spend about $31 billion in the NIH, which creates about 350,000 jobs. The important thing to remember, though, is that 85% of those jobs are not created here in Washington. They're not government jobs. 85% of that money is distributed across the country to medical schools, research institutes, et cetera. As Tim said, at Sanford Burnham, we do about $90 million a year in NIH funding, which supports a 1,200-person operation. About half our employees are PhDs. And so you get a sort of a sense of the role of this. That's about 70% of our operating budget, and it really is the lifeblood of our operation, but also medical schools and hospitals that do research, et cetera. That then creates a foundation which then encourages another $70 billion of research investment in this country alone by the biotechnology and pharmaceutical industry, which creates another over 500,000 jobs. And in addition, lays a foundation for the medical device and laboratory testing industries, which creates about another close to 1 million jobs. So altogether, it's estimated that every dollar of NIH funding generates, at a minimum estimate, about $3.5 of economic activity in the private sector. You can even make a pretty decent argument that this catalytic role that NIH funding has actually more than pays for the investment that we make. If you just look at the income tax from workers in this field and do the math, you see that from the jobs funded through NIH research, coupled with the jobs from the biopharmaceutical industry, and looking at the average wage, you can make arguments that from that alone, it drives about $60 billion in taxable revenue back to the government. So it more than doubles and pays for the investment. And that's not even considering the corporate income tax that derives from the products that are made, which is over an $80 billion proposition itself in terms of the profits that then create a taxable base as well. So that gives you a sense of this sort of ecosystem of economic development and how the fundamental research done in research institutes, hospitals, universities, primarily funded by NIH, plays such an important foundational role. I want to talk a little bit now about global competitiveness and the role of intellectual property. I think all of us believe that America is fundamentally a country of inventors. It's estimated that half of all the world's great inventions came from this country. All of us who are holding things like iPhones and cell phones can see that every day in our lives. So this is clearly the key to our economic success. I don't think we can out-manufacture the rest of the world, although certainly we could do a better job than we're doing now. But innovation is the key to economic development. And it's actually one of the places where we really excel as a nation. If you look at balance of trade issues and look at a variety of economic sectors in how much we export versus import, intellectual property is one of the areas where we do the best. In fact, it ranks number one in terms of how you look at the data, and we lead the world in that. There's nobody else even comes close to us. The next closest to us is Japan, which is 10 times less in terms of the amount of revenues that these countries receive for the rights to their patents, the ability to use those patents which drive licensing and royalty income. So this really is a key to economic development, and I have a quote here from the Premier of China that says, the future world competition will be for intellectual property rights. So I think we need to keep that in mind. So in the context of creating a biotechnology and pharmaceutical company infrastructure, the point I want to rate for you is that the inventions made from NIH-funded research in the nonprofit sector, universities, research institutes, hospitals, leads to discoveries which are patented, and those patents then become the substrate for a whole ecosystem of job creation. And you can see here that the roughly $25 billion that the NIH spends extramural leads to intellectual property, which through SBIR grants and venture capital financing, then at least it more than matches the $30 billion investment the NIH is making, and then that stimulates another $70 billion investment that the pharmaceutical companies make in this country alone. All the economic activity then generating close to $750 million in products, which become then a base for taxable incomes that help feed this whole ecosystem and sustain it. So some of the things to know about this is that when you receive an NIH grant, you're actually obligated, thanks to the Bayh-Dole Act, to file patents on those inventions and to preferentially license them to American companies, preferably small companies. 74% of all U.S. biotechnology and pharmaceutical companies have licensed patents from the NIH, from NIH-funded research. 59% say that technology is central to their business strategy. And close to 20% of FDA-approved drugs cite NIH-funded research, patents deriving from that as a source of the innovation. Interesting couple statistics. If you look at NIH grants, there are almost 40,000 that are issued across the country. 98% of those result in an invention disclosure. 56% result in a patent application, and 22%, nearly a quarter, result in an issued patent. So this funding is creating a rich patent and intellectual property franchise for our country that helps to make us competitive on a global level. Let's talk now about healthcare costs and the role of the medical research enterprise in terms of this. Now, this is something we're all very cognizant of. Right now, we're spending about $2.3 trillion, something like 16, 17, 18% of GDP on healthcare costs and rising. We actually spend 2 to 3 times per capita more on healthcare in this country than the other top 10 developed countries of the world, and yet rank 9th or 10th in most indices of healthcare outcomes. So clearly, we've got to find more efficient ways to deliver healthcare in this country. Already, we can cite a number of examples of innovations from medical research funded by the NIH and then fostered by pharmaceutical companies that have driven down medical costs. The first example I have there is an Alzheimer's drug called Namenda, or Memantine, which was invented by one of our scientists. That was approved by the FDA a few years ago for Alzheimer's. It's estimated that patients on that medicine, it delays the time from which they deteriorate to being unable to care for themselves, allows them to remain independent for about 18 months longer than normal. The estimated savings from that is over $100 billion. So the medicine actually costs the country less than $1 billion, but it saves over $100 billion by delaying the time at which these patients with Alzheimer's become dependent. The next one on the list is HIV drugs. When I was a medical student, our wards were full of people with HIV undergoing lengthy and expensive hospital stays. Now, you never see anybody in a hospital with HIV. We can treat it with a 3 or 4 drug regimen. So these are just a few of the examples of how innovations in medical research have the opportunity to drive down healthcare costs. But the opportunities are even more robust today than they ever have been, thanks to a variety of technologies that are beginning to allow us to envision an era of personalized medicine where we can make healthcare more of a science and less of an art by taking a lot of the inefficiencies and uncertainties out of it. At the heart of this is the revolutions going on in what's called next-generation DNA sequencing. It's only about a decade ago that the first human genome sequence was announced. It was a project that took 13 years and over $3 billion to get one single human's DNA sequence. Just 2 weeks ago, a US-based company announced an instrument that will sequence an entire human genome in 2 hours for $1,000. So this is game-changing technology invented here in this country, and it has the potential to totally transform healthcare, not only here but worldwide. I'll show you just a small example of some of the type of work that we're doing that could illustrate how we can extract healthcare savings from this type of technology. And I use as the illustration heart failure. There's about 500,000 Americans that have heart failure. From time to time, their disease exacerbates and they go into the hospital. If we do a good job of treating those patients and getting the right medicines, etc., they can stay out of the hospital, but if we don't, they end up back in the hospital. Readmission rates for hospitalization for heart failure cost this country for Medicare alone $17 billion annually, just for Medicare costs for that one disease. What can we do about that? Well, some of our scientists, Dan Kelly and the team, identified a genetic variation in a gene that's involved in metabolism, where some of us are born with a G at one position in the DNA code and others an A. And they did studies to look at the frequency with the patients are readmitted to the hospital. If you look over time, if you're born with two copies of G, one for your mother, one for your father, you have a very high rate of hospital readmission shown by this green line. However, if we put you on a type of drug called a beta blocker, then that rate goes way down. And so there's a real benefit to you if we put you on that type of therapy. On the other hand, if you're born with an A in one of those genes, you have a very low rate of readmission. So only a few % of those patients have heart disease that becomes problematic enough that they have to get readmitted. But if we put you on the standard beta blocker therapy, you actually do worse. So there's actually harm to putting you on that medication. So in the past, when we didn't know what to do, we're just guessing. Now with these types of technologies, we can, in a much more rational way, decide who gets what medicine and drive down the cost of healthcare. Now, genomics is not going to solve everything. Having a DNA sequence is certainly a good foundation, and eventually all of us will have our DNA sequence determined. But we're also affected by our environment. And the example I think that we can really all relate to is the current obesity epidemic, where now 30% of Americans meet clinical criteria for obese. So it's clear that we need other ways of looking at the complexities of disease. Thankfully, there are a variety of other technologies that are emerging and have been developing over the last decades, not only genomics, but metabolomics and proteomics, where we can begin to survey the various systems in the cell at a broad level. And when integrating these together, we can get a much more accurate picture of what's going on in a patient. I'll show you one example of a proteomics opportunity, and we'll talk about it in the context of prostate cancer. Prostate cancer is something that all of us men care about. It's a disease effectively of aging. Autopsy studies show that if you live to be 75 years or older, 85% of us men will have prostate cancer whether we know it or not. So the question is, what do you do about that from a standpoint of treatment and economic cost? For most men, prostate cancer is an indolent disease that they will die with rather than die from. It's something that really won't bother them. And so it's even acceptable for the right patient to use a watchful waiting approach where you don't do anything, you just sort of monitor. On the other hand, you can do surgery and take out the prostate, etc. So there have been many articles written about this in attempts to try to find out what do we do. Well, I'll just show you one example of a project we've done in my laboratory where we've looked at a particular biomarker, a protein that we discovered that binds to the androgen receptor called BAG1, and have looked at men with prostate cancer to follow what happens to them over time after prostatectomy. And what you can see here, these are basically two different ways of looking at the data, but if your tumor is positive for this marker, those patients don't do well. This is the percentage of patients still disease-free and living over time. But if it's negative for it, most of those patients live. So this just shows you an example of how you can use a proteomic marker like this to begin to, in a much more rational way, figure out who needs therapy, how aggressive should that therapy be used, and therefore be able to be able to provide that therapy in a much more cost-effective way, as opposed to sort of a one-size-fits-all approach where everybody gets a prostatectomy or everybody gets hormonal therapy, etc. And then finally, there's a whole vista of opportunities that are emerging from not looking at various analytes in cells like the DNA or the proteins or the metabolites, but even being able to study at a functional level differences in patient cells to be able to predict who needs what medicine. And I'll illustrate this in the context of what's going on in the exciting area of stem cell technologies. And I'll do that for a particular type of disease, neuropsychiatric disorders. So neuropsychiatric disorders have been difficult to study because we traditionally have not been able to get access to the brain cells from patients. Not very acceptable to go do neurosurgery on somebody and chop out a piece of their brain and figure out what's going on. But advances in stem cell technology now allow us to make brain cells in the laboratory from patients. So the way this is done is that a cell, typically from the skin, but it can be typically any cell, is synthetically converted into a stem cell through putting a cocktail of genes into it. And once it's in the stem cell state, we can then program those cells to become nerve cells or heart cells or whatever cell we need to study. In the case of neuropsychiatric disorders, which is one of the problem areas of medicine in that we don't have blood tests for diagnosing neuropsychiatric disorders, the average patient goes through 5 different attempts at medications before you find the one that works for you. And yet these are very frequent diseases. 1 out of every 6 Americans has some form of a neuropsychiatric disorder. 1 out of every 3 of us at some point in our lifetime will have a clinically significant episode. It's the largest single sector of pharmaceutical drugs. How can you figure out which medicine goes for which patient? So what we can do now is to take cells from those patients, turn them into brain cells in the laboratory, and throw drugs on those cells in the laboratory and see which ones they respond to or not. And so there's been a company spun out of the Institute called Neuromap, which is attempting to do this now, to be able to take the guesswork out of picking the right medicine for the patient and therefore drive down healthcare costs. These types of technologies also are creating the opportunity to think about even taking approved medicines that exist already that are already approved. There are around 5,000 such medicines that are approved by either the FDA, the European Union, or Japan. And using these type of technologies to see would any of those just happen to correct the defect in a patient's cell and allow us to match a medicine to the defect in a patient and to be able to very quickly then find new uses for old drugs, what we refer to as drug repurposing, which is probably the most efficient way to find new treatments for diseases. So I think I'm going to wrap up there. I have a couple slides here just with a few statistics on some of the estimates in healthcare savings that have already come out of some of the research. Just since 1995 alone, a study estimated approximately $70 billion in healthcare savings from the medical research funded by NIH, so more than twice what we currently invest in NIH. And a variety of other studies which have attempted to look not only at the directed medical costs, but the indirect costs of lost productivity, et cetera, have suggested that the funding that's provided by the NIH is estimated to save an average of $3.2 trillion annually, which is a 100-fold return on investment when you look at the amount we invest for that medical research. So the argument then would be that these types of innovations are really going to be the key to driving down healthcare costs in the long run as we try to lead the world in this new era of genomic medicine and personalized medicine. One thing I want to remind everybody is that while nonprofit organizations like Sanford Burnham, universities, hospitals do the important work of making these discoveries and helping lay the foundation, we actually don't sell anything, we don't manufacture anything, and so it is absolutely critical that those inventions find their way in the hands of investors and for-profit companies that can invest the capital needed to turn them into products that not only drive economic development and improve healthcare outcomes. And it's with that in mind that our next speaker will now tell you a little bit about what it's like to start new companies based on intellectual property that has emerged from NIH-funded research in a nonprofit like Sanford Burnham. So thank you for your attention.
As Scott's coming up, I just want to thank Congressman for coming out. Did you want to mention anything? And I know you
I like Sanford Burnham.
That's good. Okay.
I think what's been presented here is a picture of what's wrong with Washington, D.C., though. We tend to do cookie cutter programs, whether it's transportation funding or Medicaid funding or whatever. And what's being proved is, even in medicine, that's cookie cutters don't work. You can, here's your prescription, this is what you got, go take it, and hopefully you get better. And that's exactly what we're doing here. So I think there's a great analogy here.
Thank you.
All right, with that, Scott, I'll let you go ahead and get started. He's more of the entrepreneurial side of this story.
I am indeed, and I'm honored to be here. When I was called by Connect to come up and talk with the staff members, and I didn't know there'd be a congressman. I thought there'd just be staff members, but I was very honored to have that opportunity. I was given as much time as Dr. Reed, and he has a lot more to talk about than I do. But I do, what I've been involved over the last 20 years is primarily starting biotech companies. That's been my life. I'm a finance guy, I mentioned to some of you guys earlier. So I've had a great opportunity to watch the revolution in healthcare and biotech. Dr. Reed mentioned next generation sequencing in 1999 with a great gentleman who was a fantastic scientist. I know his name will be known by a lot more people over the next decade or so, but a guy named Jonathan Rothberg, who was a NIH-funded fellow, real young guy, and started a biotech company in the early 90s. He and I got together in 1999 and started 454, which was one of the first next-generation sequencing companies. It's part of Roche now. So I've been able to see a lot, and it's wonderful to see some of the applications of that technology. I did want to mention something, and I'm going to segue right. I'm going to kind of ride the coattails of your cookie cutter comment and some of the stuff that Dr. Reed said about inventions and that broken pipeline that we have. I want to mention, as a biotech entrepreneur, I've had the joy, the opportunity to employ hundreds of scientists over the last 20 years, and been involved in working with them not only to create new patents, but also work with some of the universities and license in patents that we attempt to commercialize. And if you look at those patents that you talk about, and you look at the names of the inventors, it's not, it's like the UN. So our ability to be at the leader in revenues coming in from licensing of intellectual property is a direct result of our country being in this privileged position of being such a wonderful place to learn and then to do work. And so we've been able to attract the best and the brightest from around the world. And so this cookie cutter approach to immigration, where you just say, look, we've got to take care of our program and we're just going to just cut immigration. And we need to be a lot smarter about how we attend to our immigration problem, not hit it, not use a cookie cutter approach, but be smart about it, and not be in a position where we're telling some of these people that we've trained here in this country at our great universities, our great hospitals, their NIH fellows, and then they have no ability to stay here in this country to work. So we really need to address that, otherwise we're going to lose this wonderful privileged position that we've had. Because I was talking to Dr. Reed on the way in here. In 1995, I went to Shanghai, China, to put actually it was a DNA sequencing consortium together, and Shanghai in 1995 was a bustling city, but still pretty old, and the infrastructure was pretty rickety. I was back in Shanghai in 2010, and the difference is just phenomenal. The research institutes that they've built in just the last 15 years are now equal to what we have in the US, in Europe, in terms of the infrastructure, the equipment that they have. And now you see a lot of these wonderful Chinese scientists that were trained in Europe and the US, then worked in the pharmaceutical industry, the biotech industry for 10, 15, 20 years, wonderfully experienced, now going back in droves to China. I think you're seeing the same thing in India. So we cannot take for granted our leadership position. So we really have to attend to some of the macro issues like immigration and funding of the NIH. So that was I probably have 5 minutes left now. Right? Let me talk about Sender. Sender is a little company based on some very, very exciting technology out of the Burnham. And interestingly enough, some of the science that the Sender peptides are based on is science that the NIH has funded for 25 years, some of the I'm not going to get into the details of the peptide, but some of the initial insights were invented at NIH-funded laboratories 25, 30 years ago. So this speaks to the importance of continuing to fund this research over long periods of time. So our goal at Sender is, we're talking at breakfast, we're not making drugs. We're like the old BASF commercials. We're making drugs better. So we have these wonderful little peptides, which are fragments. They're like, think of them as smaller than proteins, larger than DNA, nucleic acid sequences. They're a little bit bigger. Our goal is to use some really interesting peptides to increase the efficacy of drugs. We can increase the efficacy by getting more of the drug to the tumor, so you don't have as much collateral damage to the non-diseased tissue, the healthy tissue. And we can decrease toxicity by allowing the physicians to use a much smaller dose, because the drug tends to concentrate in the tumor, so you can use less drug. We can also use this peptide for imaging support, so when you go into the physician, we can use this peptide to really let the physician see where your tumor is. And the interesting thing about this peptide is it's a platform, really, that we can use not only to make small molecule drugs better, but also antibodies better, nanoparticles better. So we can take existing drugs and future drugs and really make them a lot more efficient at killing tumors. So the company, again, spun out of the Burnham. We completed the license in July of 11, so just about 7 months ago. The company was founded in 2010 based on some, this idea of working to get this technology out of the company. It took a long time to license from you guys. So not a ding on the university. I think not university, but the institute. I think they had some stars in their eyes that they were going to try to sell it to a pharmaceutical company. Yeah, exactly. So I was competing with a lot of people. So we decided when we got that technology that they had done enough work, NIH-funded work, that we weren't going to go run out and get venture money to fund this thing. We were going to try to continue to leverage that NIH-funded money, or NIH funding, with additional NIH grants. And so the NIH makes wonderful grants available to small companies through there's the SBIR grants, there's START grants. And so Sender's been very, very fortunate. We have about $2.5 million in grants that are specifically focused on translating research out of institutes like the Burnham into clinical practice. And so we're using this money to support an IND filing, which is the filing that you have to make to start experimenting with your drug in humans. We expect to file that IND in the springtime and dose the first patients in the summertime. We also got a nice grant to fund some preclinical pancreatic cancer work, and if you guys are familiar with pancreatic cancer, it's one of the worst possible cancer diagnoses that you can get. It's a cancer that moves very, very rapidly and is pretty uniformly fatal. So I have 2 slides on technology, little cartoons. I am going to skip right to this next one. It's a cyclic peptide, binds to a receptor that is highly upregulated on cancer cells. So that's where the homing portion comes in. And then through a process I'm not going to explain here, it initiates a bulk transport mechanism. So what it does is it says to the tumor, it triggers the tumor to be very acquisitive to everything in the circulation around it. Typically tumors are exactly the opposite. They're trying to keep stuff out and try to live. They want to get nutrients and so forth, but they want to keep drugs and other things out. So this tricks them into just pulling everything in. And so one of the really interesting things from a regulatory standpoint is we do not have to link the peptide to the drug. If we had to do that from a regulatory standpoint, we have a brand new drug.